So our next speaker will be Simon Tannock, who will tell us about opportunity to use diatoms to control cyanobacterial blooms. So over to you, Simon.
Okay, it appears that the presentation is mixing up my graphics, but that's okay, we'll live with that. So I'd like to acknowledge the co-authors who are from the couple of the utilities that I work with in gathering this data. And so I'm I am a co-founder and from AlgaEnviro, and we are a manufacturer of a micronutrient mixture that we use to enhance diatom growth. So fortunately in this room I don't think I need to go through in too much detail obviously the issues that are faced by people who are trying to manage water or look after or treat water, in that nutrients and algae are usually the two biggest problems. Both in the small scale and in the large scale, and these are boats in Lake Erie, just to give that some scale. So depending on whether it's a natural lake or aquaculture or wastewater treatment, then algal blooms and macrophyte growth can cause a number of different issues. So with the way our system works, we basically go all the way back to the Redfield ratio, which I'm sure most of you will be familiar with, where in photosynthesis there is a ratio between the amount of nitrogen and phosphorus that's used, and obviously iron and a lot of other micronutrients as well. And in a eutrophied system, I've kind of mixed up the ratio there a little bit. So if you imagine that your iron concentration is the same, then by it becoming eutrophied, that means that the ratio would be much, much higher for nitrogen and for phosphorus. And so we're looking with our system to address that. And the way we do that is that we have looked at a large number of micronutrients that are consistent in different concentrations across quite a range of different algal recipes for growing algae in culture, and we then have attached that on basically what is the skeleton of silica in a gel. And so when it's in the water, it looks like silica to all of the algae, and that means that it's only taken up by those types of algae that would require silica. And I will just touch on and stress that the product diatomics is just micronutrients and silica. It doesn't contain diatoms, because we regularly get asked, well, what species of diatoms are you adding, or can we have a truckload of diatoms, please? And we don't do that. We are relying on the natural populations of diatoms in the water to correct themselves, so we're not creating issues of introducing new species where they're not used to being. So again, with this crowd, I probably don't need to go into too much detail about diatoms. Obviously, they're pretty well found everywhere, and they do have a requirement for silica. And going back to that Redfield ratio, they use silica on basically a 1-to-1 ratio to nitrogen. And the cool thing about diatoms is that whilst they are taking up nutrient, they're also at the bottom of the food chain. So they're down here in this picture, and we've seen a number of different cycles and systems for water and nature today. So by growing diatoms, you are then feeding these animals all the way up the food chain. So if through the addition of diatomics you can draw down nitrogen and phosphorus as the diatoms bloom and then they're grazed, you are moving that nitrogen and phosphorus into the animal ecosystem and then up those higher trophic levels. So this is just a sort of a stylized drawing of that same system. So by adding diatomics, we grow diatoms, and then it moves all the way up the food chain. And we can, this occurs both with cyanobacteria, but also a lot of macrophytes. So we have done work with different aquaculture and council parks and things where they have macrophyte or Zola issues or filamentous algae issues, not cyanobacteria. And in each of those, we can reduce the ammonia and nitrate profile and then the growth of these other algae or plants. So I've got 3 case studies that I want to go through very briefly, and 2 of them I presented 2 years ago. This first one is one of them. Before we got started with them, it looked like that, which was around about 2.75 million cells of blue-green algae. And well into a year later, it looked like this, and so it was considerably healthier, and we had just over a 99% reduction in blue-green algae in the first season, which is that data there, and that's what I presented last time. Subsequent to that, they have been taking more cell counts. They are now at the stage where they actually only take cell counts when they really feel like there's something worth looking at. They don't do it as a routine anymore because of the fact that they haven't been seeing as much blue-green algae. They did have one little blip there. They noticed that things were looking a bit strange in the lagoon, so they went down and checked out the dosing machine, which is a solar-powered automated system, and the battery had died. So dosing had stopped, and there was quite quickly a response in the blue-green population picking up there, and then once they replaced the battery and everything got dosed and started again, then it all started to settle back down. Those points where the arrows are all samples where there was a 0 blue-green algae cell count within that, which in a treated effluent site is quite astounding to me at least, because we haven't seen that before, but this site has performed very well. This is a raw water storage on a pig farm, so they were just drawing from their farm dam, and if they got to the point where they had too many blue-green algae, they would have to go on to town supply. So they started using our system because the treatment through using diatomics is for a year was less than 1 month's worth of town supply, drinking supply. The green line up the top here is the period where diatomics was being dosed. They were under very tight budget, so they used manual dosing, not an automated doser. And just around that November period, they rang me and said, "Oh, things are going quite blue-green again. We need to, up the dose." And I said, "Well, what is your dose on, and who's doing it?" And they said, "Oh, it's on, whatever. I can't remember the number actually, but they said Dave's doing it." "Oh, hang on, Dave's on holiday." I said, "Well, who's doing it?" And they said, "Oh, nobody's doing it." And I said, "Oh, well, there's the answer then. That's why it's doing it. Start it again. Somebody else can be Dave for a while." So they restarted it and took a sample, and there was a continued peak bloom there, and then it started to come down. The other thing I should also highlight is that the blue-green and total cell counts are here, and the diatom numbers are over here. So even though you might see 70,000 in this site for total and 45,000 for blue-green, our peak in diatoms was only 5,000 cells per mil. And that will come into a bit later in my discussion. On the same site, on a different dam, Dave wasn't in charge of the diatomics blooming, and so we didn't see that same peak. So I believe it was due to the fact that the dosing stopped, not just because of the conditions at the time on that site. Our third case study, again, this is data that I presented last time, and when I asked for the data for now, they said, "Oh, we haven't been monitoring it again since this got started because it's been looking so good, so we didn't feel we needed to," which obviously I'd already submitted my abstract, so I was a bit concerned about what to do about that. But again, just looking at those numbers, when we started with them, they were at 7 million-ish cells of blue-green algae, and our highest peak there was about 22,000 of diatoms. But during those dose periods, blue-greens came down, and then diatoms at some point would come up, but not always that high. But the data that they did have, which I think is really quite interesting, is the reason, there goes those graphics again, that's fantastic. The other issue that they had, and one of the things that they were hoping to resolve, was the pH within the lagoon, because at times it was spiking to nearly 10, which creates real issues for them for their releases. So since being on diatomics, there is a consistent trend downwards to more sensible levels around that 8.5 to 8. And I think part of what's happening there is that the microinvertebrate and the invertebrate and vertebrate profiles have changed considerably. So 8 weeks after they started dosing this dam, they rang me and said, you should come down and have a look. There is just insects everywhere. And after 12 weeks, they said, for the first time in 5 years, we've got birds feeding here. And the turtles are walking from other parts of the farm and coming and feeding in this area. Now, I don't know how turtles 500 metres away know that there's a dam that's doing really great things, but... Sorry?
Birds told them.
Birds told them. Okay, good to know. Especially those turtle-eating birds. So, but anyway, they've said it's just been this continuing increase in health and biodiversity. They're seeing species of birds nesting there that they've never seen before as well. And so, again, this comes into touching on a bit more later. So, in thinking through a number of the things that we've seen in the data, particularly about those very low diatom cell numbers, and yet still seeing, good results in all of this, we got to realising that we were probably growing a lot of benthic or biofilm periphyton type diatoms and not so many suspended diatoms. And so also thinking about wetlands and mangroves, we know that they have large surface areas and they're also very good for creating habitats for all of the whole food chain, which, as I highlighted in that earlier picture, was that we want diatoms to grow, but then we want the grazing pressure of all of those animals to do that. And most of those animals need somewhere to live and to have that activity and lay eggs and all those sorts of things. So particularly in large reservoirs where it's just water for most of it, or in constructed water bodies such as treatment lagoons where it's a plastic-lined pond, there aren't habitats for the sorts of systems that we're trying to encourage to live. So all of that biofilm activity can happen if you have a great surface area, but we don't always have those great surface areas. So I'm going to have a go at running a video now. And this is from that case 3 system. And if it starts, I'll start talking about it, and if it doesn't, we'll move on. Oh, there we go. So there'll be blue rings pop up here, and when that happens, there's an insect growing, an insect popping up, taking a breath and dropping back down. And that's some of the insect activity that these guys were telling me about. And so it's quite a brown predominated biofilm and flock in there, and then there's insects just popping up and down all the time, and then there are obviously other animals predating on them. So that's kind of where we got to about thinking, how do we drive this harder and make that work more efficiently? Oops, and it's going to go again. Right, so by thinking about those biofilms, I needed to try and figure out how to get a structure that would grow biofilms in water and stay there all the time. So we've been working with a company that produces a media that has a very high surface area, and there are all sorts of nooks and crannies and things for invertebrates and others to live in there. And if we then create that into a structure that can be pulled out across a lagoon system, then that allows a much larger biofilm system, and then obviously all of the habitats where we can get this activity to grow. So this is a photo of one of those systems in water after just for a few hours. So it's already obviously developing some air bubbles and things down in here. And in a matter of hours, you'll start to get some biofilm growth on there as well. For reservoirs and much larger areas where it's a kilometre wide and you can't put a rope from one side to the other, then we are developing ring structures, a bit like salmon sea cages. These will sit at 1/2 metre below the water with the loops of rope, and I actually have brought some with me in case anyone wants to get an idea of what this stuff looks like. And and then the water can just flow through these. This system we installed last Tuesday just as a bit of a trial at one of our sites. I received an email with video yesterday of that site, and the ropes have sunk down a bit better and it's all settled in, but there's already a lot of fish activity. You can see water flicking up and the ropes shaking and all sorts of stuff, and they've said it's just crazy in there. You can see all this fish chasing around. There's carp in this water. So even though I don't think that whole trophic cascade has developed really well, it tells us that biofilms are developing. It's working as an animal accumulation device, and as those diatom biofilms continue to grow, that's, you know, the result that we're looking for and have designed this for is going to start to happen. So that's just a recap on the data I presented last time, with obviously that longer perspective that we are continuing to make positive changes within the water. We're continuing to see, both at these sites and other sites we've treated, reduced blue-green algae, stable systems, and now we're moving toward these biofilm floating habitats to increase the stability of the biology, allow the diatoms to stay in the photic zone so that even in a stratified system they won't drop down, and we can solve a biological problem with a biological solution. Thank you.
Thank you for that very interesting presentation. We have, again, plenty of time for questions. Please wait for the microphone. We'll start at the front here.
What sort of impact did it have on dissolved oxygen concentrations in these different systems?
Generally, so I'll maybe speak to a lot of the data we have. They don't measure things like that because it's not really of key interest to them. One site that we have dealt with is an aquaculture site, it's Barramundi Farm, so obviously it's of great importance to them. In the afternoons it might get around to the 9 and 10 milligrams per litre, and by morning it's around 2 to 2.2 as a low. They were always getting very upset when it was going below 2, and they're not seeing that now. They've said it's just securely above 2. And I think it's because whilst we are creating diatom blooms that are doing this activity, they're being kept low by the animal activity that's grazing onto them. So we're seeing changes, but not these massive fluxes.
Okay, so by concentrating the carbon from the, I'm going to think of this in very simple terms,
sure.
The carbon from the phytoplankton in higher trophic levels, they respire less than if you just had bacteria respiring the phytoplankton themselves. So that's attenuating the diurnal range of the dissolved oxygen concentrations. I'm just trying to figure out.
Yeah, I think the carbon thing is that there are more animals there, so they're producing an increase in CO2. That obviously will mean that the pH comes down slightly. The diatom bloom or the algal bloom isn't as large, so whilst they're busily producing oxygen, there's not so many of them that they're causing massive draw up and then all sitting there to pull it all back down during respiration. But that's certainly an area of, if you wanted to get to that nitty-gritty to understand, that needs to be researched. But that's the behaviour, is that we don't see massive drops of DO or massive spikes of DO. And that's my logical explanation for it.
In theory, it's a good plan. I mean, pH is going to be within a narrow range, DO is going to be a narrow range, and like Brad mentioned, you're making nutrients into edible something sounds all good. But we are in the hands of God, or nature's hand, when you apply various things and hope that the diatom we like or you like would be popping up. What kind of control we have, which diatom that's going to come through, and if we get a nasty diatom that we don't like pops up, what exactly is going to happen, and how are you going to control it? The reason for the question is the reservoir we have in mid-north coast, we have diatom growth during summertime, and we don't have a treatment system. It's a dosing system, and diatoms is a mess because chlorination is poor and things like that. again, so it's multiple, right? One, what diatom we can control, and if we can control to a certain diatom, probably we can try in a even drinking water reservoir, so we can have a preferable diatom to live there. Do we have a solution, or is it luck?
It's both. So possibly it's luck, but I give the analogy that if you put a case of beer in the middle of a rugby club rooms, one rugby player isn't going to be the one that gets all the beer. And so everyone's going to get some. So if you put diatomics in the water, I think that all diatoms will respond, and it will probably open out the types of diatoms that will grow. Whereas if you're seeing one sort of species growth, it might mean that the conditions are such that others won't grow there. But if you make it really suitable to all diatoms through this way, then I would expect that the breadth of species would increase, and then draw down the dominance of that one species. But until somebody wants to do that study under those conditions, I can't answer that beyond rugby club room analogies.
Thank you. Thank you, Simon. Sorry, we're going to have to move on, so please ask Simon some more questions afterwards. Yeah, and the nice thing about your rope is that it doubles up as a Christmas decoration. So here's that. Yep.